Hello everyone, I'm Yasamin Tabatabaee from the University of Illinois, and in this talk I'm going to present Quintet rooting, which is a method for rooting species trees under the multi-species Coalescent model. This is a joint work with Kaushika Sarkar and Tandy Warno. Rooted species trees have multiple applications in biology. For example, they're useful for studying adaptation, biodiversity, phylogeography, and coevolution. However, there are still several challenges in finding the correct root in many biological data sets, and most species tree estimation methods produce unrooted trees, and therefore additional methods for rooting these trees are needed. So the problem that we are studying here is finding the position of the root in a given unrooted species tree. Existing methods for rooting species trees can generally be divided into different groups based on their assumptions about the input data. Perhaps the most commonly used technique by biologists is outgroup rooting, where a species that is known to be distant from the taxa in the input set is added to the set, and a tree is estimated on this expanded set, and the edge that divides this in-group and outgroup determines the position of the root. But selecting an appropriate outgroup can be challenging for some data sets because of, for example, issues such as long branch attraction. Another group of techniques that are used are distance-based methods that use the branch lengths of the tree to find the root based on a specific optimization criteria. These methods are mostly based on molecular clock analysis or non-reversible models of DNA substitution, and most of them are sensitive to deviations from the molecular clock. And finally, there are gene-based methods that take a set of single-copy or multi-copy genes as input and then use these to find the root in an unrooted species tree. Examples of these methods include a stride, which finds the root based on gene duplication or loss events, and a site-based method by Tiana and Kobatko that is relevant to rooting trees under the MSC, but assumes a strict molecular clock. It's also good to note that outgroup rooting and distance methods can be used for rooting both gene trees and species trees, but in this talk we will only focus on rooting species trees. Also, generally, most species tree rooting methods don't account for the biological processes that cause discordance between gene trees and species trees. So this work is in the context of phylogenomics, where the goal is to infer a species tree given a set of genes that are recombination-free regions across the genome. And usually this species tree estimation is complicated by processes that cause discordance between gene trees and species trees. There are several causes for gene tree discordance, for example, incomplete linear sorting, gene duplication or loss, and horizontal gene transfer. Here we will exclusively focus on ILS, which can be modeled by the multi-species Coalescent model or MSC. MSC models a random process that describes the way alleles coalesce in populations, and when two lineages fail to coalesce to their common ancestor, it results in incomplete linear sorting that creates the opportunity for gene tree discordance. It's a well-known result that under the MSC, for 4 or more species, the unrooted topology of the species tree is identifiable from the probability distribution that it defines on the unrooted gene trees. This theorem is proved in a paper by Allman, Dayton, and Rose, and henceforth I'll call them ADR. A key that leads to this result is the observation that for 4 species, the most probable unrooted gene tree has the same topology as the species tree. And this means that if we are given the probabilities of the 3 possible unrooted quartet trees, the dominant one will give us the species tree topology in these 4 taxa. And this result has been the basis of several quartet-based species tree estimation methods such as Astral. There is another theorem in the same paper by ADR that shows that for 5 or more species, the rooted topology of the species tree is also identifiable from the probability distribution of the unrooted gene trees. ADR derived a set of linear invariants and inequalities on the probability distribution of unrooted gene trees, and they proved that these inequalities and invariants are sufficient to identify the rooted species tree topology. And this means that if the probability distribution of the unrooted gene trees is known exactly, there will be just one rooted species tree topology that satisfies all of these invariants and inequalities. And interestingly, this result has not been used in any species tree estimation or rooting method before. In order to better understand this result, we need to first look into the properties of quintet or 5-leaf trees. On 5 taxa, there are 105 different rooted binary trees and 15 unrooted binary trees. And each unrooted 5-taxon tree can be rooted on any of its 7 edges. So for example, on the left side of the figure, we can see an unrooted 5-taxon tree where its edges are marked with numbers, and rooting it will result in one of the trees on the right. And generally, the topology of rooted 5-taxon trees can have 3 different shapes, which are named caterpillar, balanced, and pseudo-caterpillar. The invariants and inequalities found by ADR define a partial order and a distribution of unrooted gene trees for each model species tree. And the partial order of each tree can be shown with a Hassett diagram, as we can see an example in this figure. In this talk, I will show the 15 unrooted 5-taxon trees with T1 to T15 and their probabilities with U sub I values. So here we have a caterpillar tree in its Hassett diagram. Each circle in the Hassett diagram defines an equimulus class, meaning that all the gene tree probabilities inside that class need to be equal, and this defines the set of invariants. For example, U4 equal to U13 in class C4 is an invariant. Similarly, each directed edge shows an inequality where the class at the source of the edge must have strictly greater values than the class at the target. For example, U4 must be strictly greater than U5, and this defines an inequality. An important thing to note is that this Hassett diagram is independent of the branch lengths of the model tree. However, the equivalence classes that are not related by an inequality, for example here C3 and C5, can merge for some values of the branch lengths. According to the ADR theory, each of the 105 rooted binary trees correspond to a unique Hassett diagram, but the shape of this diagram only depends on the topological shape of the tree. And we can see examples of these Hassett diagrams for each of the three topological shapes here. And ADR proves that given the unrooted tree and the true distribution and the UI values, there is only one partial order out of the partial orders corresponding to the 7 possible rootings that is consistent with the true distribution. So we wanted to use this identifiability result to develop a method for rooting species trees from unrooted gene trees. The first question that comes up is how can we infer the model species tree when we are given the estimated gene tree distribution? Clearly, none of the invariants will actually hold since we have estimated data. And even the inequalities can be reversed when probabilities are very close, which can be due to quality of the data or values of the branch lengths. And in practice, we always have gene tree estimation error that adds noise to the distribution and further distorts the gene tree probabilities. To formulate this as an optimization problem, we consider the penalty for each invariant and inequality in a partial order that is violated in a given distribution. So, for example, on the left, we have the estimated probability distribution on the 15 unrooted gene trees, and on the right, we have a caterpillar model species tree with its partial order. We want to see how well this distribution fits the relationships defined in this partial order, and if it doesn't fit, to what extent does it violate the expected relationships. A violation for an invariant is when two probabilities that must belong to the same class are unequal. So, for example, here U11 and U14 must have equal probabilities under R according to the partial order, but they are different in a distribution. Similarly, a violation for an inequality is when a probability that must be less than another probability is actually greater than it. So, for example, U12 must be less than U9 according to the partial order, but the relationship is reversed in a distribution. So we define a cost function based on a linear combination of these invariance and inequality penalty terms that are specific to a rooted tree to measure the fitness between a given distribution and the tree. And this cost function is defined so that given an estimated distribution, it's minimized for the model tree whose partial order best explains this distribution. We selected our cost function based on a set of experiments on simulated data sets by comparing a few different functions that were all linear combinations of the invariants and inequalities and differed in terms of the number of penalty terms that they were using and the weight of each penalty term. The details of these experiments and its results were provided in the paper, and here we can see the final cost function that we selected. So in designing this cost function, we also noted that different tree shapes can have different numbers of penalty terms, and therefore the fitness cost might become biased towards a tree shape with a minimum number of penalty terms, which we call category bias. And therefore we use some normalization factors in a cost function based on the class sizes to somehow balance the number of penalty terms between different categories, and we observed that this can help avoid this bias in practice. So this led to the development of Quintet routing. I might also call it QR henceforth, which takes as input an unrooted species tree T and a set of unrooted single copy gene trees on the leaf set of T, as well as a cost function, which we saw an example of it in the previous slides, and returns a rooted version of T that minimizes the cost across a selected set of quintets. For the base case of rooting a 5-taxon tree, QR first estimates the unrooted gene tree probability distribution from the input data, basically the UI values, and then for a given cost function, it searches all rooted versions of T to find a tree that minimizes the fitness cost. For rooting a larger tree, QR first selects a set Q star of quintets of taxa in the leaf set of the input tree, where the size of this set can be at most n choose 5, and in a pre-processing step, it computes the cost for each rooted version of each selected quintet. It then searches all rooted versions of T and returns the one with the least sum of the cost across all quintets in set Q star. So the runtime of QR is O of k, the size of Q star, which will become O of n to the 5th k when the information from all of the quintets are used, where here n is the number of species and k is the number of gene trees. O of n to the 5th k can become quite large when the number of species increase, and therefore we explored an approach for reducing the size of this set so that it changes linearly with respect to the number of species, and we call this QR with a linear encoding. So in this encoding, for each edge of the unrooted tree, if this edge was adjacent to a node, it partitions the set of taxa into 2 subsets, as we can see in the figure, and we form a quintet for this edge, ensuring that this node and at least one taxa from each of the subsets are picked. And if the edge wasn't next to a node, it divides the set of taxa into 4 subsets, and we pick a quintet by ensuring that at least one leaf from each of these sets is picked. So this reduces the runtime of QR to O of nk, and it also has the good theoretical property that the root of the tree is identifiable from its unrooted topology and the correct routing of the quintets in this encoding. For our experimental study, we use both simulated and biological datasets. We use the mammalian simulated dataset as our training data for designing the cost function, and an avian simulated as well as an avian biological dataset as our test data. Our test datasets had varying amount of gene tree estimation error and number of species, and ILS level was moderately high. So we compared 4 different rooting methods with QR, basically the leading methods that existed for rooting phylogenetic trees that could be applied to these datasets. And we used these routing methods to find a root on both true unrooted topology and an estimated unrooted topology that was produced by Astral. And finally, our evaluation criteria was average clade distance, which is an extension of Robinson-Falls distance for rooted trees. And now we will see some of the results from our experimental study. Here we can see the results of an experiment for rooting the true species tree topology for varying number of species, starting with only 5 taxa. And the dataset used here is an avian simulated dataset where the sequence length is 1000 base pair, which results in about 40% gene tree estimation error on average. So the y-axis shows the average clade distance, which is a measure of error, and so lower is better, and the x-axis shows the number of species. And the box plots show the average error of the routing methods, including QR with a linear encoding, which is in green, a root digger, which is in blue, and the other 3 methods are MAD, min, var, and midpoint, which are distance-based methods. We can see that for the case of 5 taxa, QR has the lowest error, followed by root digger, and then the distance-based methods are generally very close, but they're worse than these 2. For the case of 10 taxa, all methods have lower error, but QR has the sharpest drop in error, and in this case, the distance-based methods perform better than root digger. The trends for the rest of the number of species is similar to the case of 10 taxa, but one thing that we can note is that most routing methods benefit from denser taxon sampling, and therefore they improve with an increase in the number of species. And QR with a linear encoding has the minimum error across all numbers of species, but its error becomes stable after some point. And here we can see a comparison between routing methods for routing a tree estimated by Astral for 10 taxon avian datasets for varying sequence lengths, which results in different gene tree estimation error rates from 0, which is for true gene trees, to about 70%. And the methods are as the previous slide, except that we've also added the default version of QR that uses all quintets as well. The trends show that QR with all quintets has lower error than QR with a linear encoding, but they generally have very close error, and they both outperform other methods, except in a model condition that has the highest rate of gene tree estimation error, where they are in the middle of the pack. And the distance-based methods appear to be better than root digger in all model conditions in this data set, with midpoint routing slightly outperforming the other 2. And we can also see that QR is more affected by gene tree estimation error than the other methods. So to summarize, QR is an ISMB approach for rooting species trees. It's based on a theoretical result by ADR that proved the identifiability of 5,000 rooted trees from unrooted gene trees. And in the datasets that we studied, it has shown better or comparable accuracy to existing methods, except when gene tree estimation error is very high. So this study suggests several directions for future work. For example, methods for rooting species trees could be explored on datasets with more model conditions, possibly with other causes of gene tree discordance, and also larger trees. We did not establish a statistical consistency for QR, which is an important consideration. And finally, the ideas that were used here for rooting an unrooted tree could be used in developing species tree estimation methods that produce rooted trees directly from unrooted gene trees. And in the end, I would like to acknowledge my co-authors, members of the Warner Lab at UIUC for their feedback, as well as funding and competing resources. Thank you for listening, and I look forward to having your questions.